I'm Kara. I'm hosting today, and I'm thrilled to introduce our speaker for the Living Earth Collaborative EECD seminar. Today we have Dr. Albert Soewongsono. Albert is an applied mathematician who is interested in developing models for the evolution of species. Currently, Albert is a postdoctoral research under the Landis Lab. He told me he's been here about a year and is really liking his time here. And he's working on developing mathematical models for species evolution that consider species interactions with their changing environmental conditions. His research interests primarily lie in the application of stochastic processes to model evolutionary processes in species-level phylogeny. In the past, he has developed evolutionary models in which their process depends on different factors such as age, species age, traits, and spatial information. So please join me in welcoming Albert.
Thank you, Kara, for the nice introduction, and hello, everyone. So my name is Albert. As introduced, I'm a postdoc in Landis Lab. I'm an applied mathematician by training, and today I'm going to present this talk about different models in for macroevolutions. So like I said, I'm a applied mathematician by training, so I'll try to explain my talk as clear as possible, but to all the biologists here. But if there is anything unclear during my talk, feel free to interrupt me, and then I'll be happy to explain. All right, so just as a general outline for the talk, so I will go through the introductions, and then I will basically go through by presenting some classes of models for studying macroevolutions. So I'll talk about age-dependent diversification models, and also trait-dependent diversification models, and last is the diversification models for species range evolutions. So as an introductions, let's start by looking at these simple phylogenetic tree with 4 extant taxa. So phylogenetic trees basically describes relationships among evolutionary lineages. So as you can see here, I have some makeup tree consisting of, say, you have the elephants there, and then you have armadillo, you have the monkey, and then you have tiger. And basically all these branches describes how far these species are related to one another. And I'll say that phylogenetic trees are used everywhere. I will go through that in the next slides. So for example, the obvious applications that phylogenetics are used for studying species diversifications. As you can see here, you have these large empirical phylogenies that describe angiosperms, and then you have the group of avian species. But phylogenetic trees can also be used for species conservation. So this paper from 1994, it basically computes phylogenetic diversity for a given phylogenetic trees. Then from this measure, it's used to basically study which species to be preserved. Phylogenetics are also used for virus diversifications. So this image over here is basically describes the SARS-CoV-2 mutations in Asia since the start of the pandemic. So you can also use phylogenetics. There are some non-obvious applications for phylogenetic studies. For example, over here you have the application for the using phylogenetics in linguistics. So these research is basically describes diversifications of Japonic languages in Japan. You can also use phylogenetics in folklore. So you have these phylogenetic tree that describes diversifications of different variants of the Little Red Riding Hood stories. So you can see that there are various applications for using phylogenetic trees. Before I go into the modeling part of the talk, I'll go through first by talking about the components of the phylogenetic trees. So you start by having these roots. So this is your basically your common ancestor of all the currently living species that you consider in your study. And then you have these internal branch that basically connects between 2 internal nodes. So internal nodes here is basically the ancestral species of your currently living species at the present. And then we call these species at the present as tip nodes or leaves of the tree. And then you have these external branches that basically connect your ancestral species, your internal node with your external node, with your tip nodes. And phylogenetic trees comes in different shapes. You have these kind of balanced balanced tree, but you can also have other shapes like this imbalanced tree. So what we want to know is basically studying about different factors that may maybe influence the different tree topologies that you can have. So what you can do is basically you need a mathematical models that studies for that accounts for lineage diversification. So one example that you can use is basically you can apply a birth and death model. So this is where the mathematical models come into play for phylogenetic inferences. I would like to make a statement that no models can capture the true process underlying empirical data because there are some unknown factors that you have from your empirical data that cannot be accounted from your phylogenetic models, from your evolutionary models. But that doesn't mean those models are useless. They can still be useful for testing hypotheses and improving rate estimates. Say you have like two models that basically try to answer same thing, and then you try to find which model that better fit to your empirical data that you have. So there's a famous saying that all models are wrong, but some are useful. All right, so what we want to do here is basically we try to solve the impossible. So here I have these equations, these mathematical equations. So this equation basically describes a process from a diversification model. So these sets of equations, they don't have an analytical solutions, meaning there is no closed form expressions that describe the solution for these equations. And as you include more and more features in your model, this equation can become more complicated. So like, for example, like this one here. So definitely you need like an alternative approach in order to solve this impossible. All right, so let's talk a bit about the tree generating process, how we can simulate trees forward in time. So here I'll consider the species phylogeny. So the events here will correspond to events that shape evolution of species. So let's start with so you have the root and then you have the first internal node, and then time goes from the top towards the bottom. So the past towards the future, towards the present. And then you have your first speciation event, basically your splitting event where your ancestral species give rise to two daughter species. And then during the lifetime of these species, the species can undergo like a state transition. So you can imagine these traits as being like, for example, maybe like a habitat types or maybe like a food preferences. So during its lifetime, it can basically transition between any of these states in your character, in the species character. But you can also experience what we call as an extinction event, where the branch just stops there when you have an extinction event. So these are the three different events that basically form species phylogeny. So you have the speciation events, extinction, but you can also have a branch that survives till the present time. Yeah. So what's the mathematics behind this diversification process, this linear diversification? So let's start by having these, again, this simple tree just illustrates the concept. So now I have a phylogeny tree with two extant species and then one extinct species denoted by this red node. And then what we want to do is basically we want to basically measure the waiting times for each of these events that you have, like these speciation events and then these extinction events. So let's start with this blue line over here. So this blue line over here, you can interpret that as a time for which a new species persists until its next speciation event. So just until the splitting event, the speciation events. So you can basically draw the waiting time to that next speciation event, say draw from some probability distributions. So you have this probability density f of t. And same can also be said for this red branch that describes the extinct lineage. So this is the waiting time until species went extinct. It can also be drawn from, it doesn't have to be the same probability distribution that describes your speciation events. It could be like a different probability distribution, so you assign for that. Now the process that occurs on the black branch is a bit as a different representation, because this is the branch that survived until the present, meaning we don't know what could happen to that species. It may undergo a speciation events in the future, or it may go extinct in the future. So this is basically the probability that that branch being survived up to that present time. So you can basically use the survival probability to draw the time. So now I talk about having like two different phylogenetic evolutionary models, and then way of actually choosing which one is a better fit, a better model to the empirical data that you have. So what we can do is basically you can use statistics. So you can compare different summary statistics between your what you expect to get from your model, your simulated data, and compare that with the empirical phylogeny that you have. So we can categorize these three-shape statistics by looking into, like, for example, its topology, so its shapes. So here I have like two different tree topologies. I have the imbalanced tree on the left, and I have the balanced tree on the right. And then there are various, and then these three topologies basically could be used to indicate evolutionary patterns among your species. So these are different tree topology statistics that people use. So you have this beta statistic, and then here you can see these two phylogenies. They have different beta values. One is a negative, the other one is positive, and this is the definition for each of these, the meaning of each of these range values range. We also have this Collis index people use, and also the second index. So these are some commonly used tree topologies statistics that people use to measure the balance of the tree. But you can also have basically statistics that measure the distribution of your nodes, your internal nodes across your phylogeny. So how the nodes distribute across your tree. So you can have, maybe I have trees that looks like this on the left, and then you have trees that looks like that on the right. So they all tell a different story. So this one on the right, you can see it has a longer branches towards the presence instead of towards the roots. So all your speciation events tends to accumulate in the past rather than in the present time. So what it means is that you have a decrease in the diversification rate through time, because your speciation events are more likely to occur in the past. But what you can see from the right-hand side is that it's the opposite of the left-hand side tree. So now you have longer branches towards the root instead of towards the present. So you have an increase in the diversification rate through time, because your speciation events tend to accumulate towards the presence. And then this is the statistic that people use to basically describe these distributions of nodes across the phylogeny. So how do we improve the diversification model? So say you have your empirical data that you observe from your present day species. Now you have a model. So you have your empirical phylogeny. If you have a model, you can define what we call as a likelihood, tree likelihood. So this tree likelihood basically describes the probability of observing this tree given model parameter and given your model parameter. Now, if you have a likelihood functions, you can do a statistical inference method, maybe using like a maximum likelihood to actually get rate estimates based on your model parameters and then empirical data that you have. Now, what we want is basically we can try to improve these rate estimates maybe by having more realistic model. By having more realistic model, I mean that maybe we can include more features from your species. So maybe say you want to incorporate species age into your rates, or maybe your species traits into your rates, and etc. That's where I go next by going through like these different classes of models. So start with the age dependence diversification model. So what is an age dependent model? So age dependent model is a lineage diversification model where rates basically depends on your species age. So your speciation rates and your extinction rates all depends on how old the species are. And the species age is defined to be like an elapsed time since the species arose until the next speciation events in your tree. So for example, here I can assign age of 0 at the root, and then all the way up to like a T1 before it undergoes the speciation events. Basically, if I have like this so-called symmetric speciation, once you have a speciation event, you have two descending lineages following that speciation event. So what you do is basically you restart the process. So you restart the age of these two species at 0 again. So now it starts from 0. But if you can also have different scenarios. So for example, this kind of speciation, you have an asymmetric speciation where basically following a speciation events, one one branch is basically a continuation of the ancestral branch. So it still accumulates the species age from the ancestor, but the other one is basically a new species. So you assign an age equals to 0 for that lineage on the right. So what are we actually to find? We are trying to find using this age-dependent model, where we'll try to answer a question like whether you tend to have this scenario where younger species are more likely to speciate or maybe becoming extinct, or you tend to have this scenario here for your data where older species are more likely to become to speciate, for example. And then whether we can actually improve rate estimates given empirical data by incorporating species age into your rates. So these are some empirical studies that actually found that incorporating species age into your rates actually improve the rate estimates. So this one, they applied their age-dependent model for an empirical phylogeny, and then they found that they have a better rate estimates given if they have an age-dependent model compared to if they don't have a age-dependent model, where rates don't depend on the species age. So before I go further into the modeling part, let's pause a bit and then talk about rates and also waiting times. So maybe I think everyone's maybe familiar with these exponential distributions. So these exponential distributions, it models the waiting time until an event occurring. So it's mostly described by this parameter lambda. It can take any value, any positive values, and then lambda basically is defined to be average number of events occurring in 1 unit time. If I take 1 over lambda, it's basically the average waiting time between 2 consecutive events. So just an illustration, if I have lambda equals 0.5, the expected waiting time is 2 unit time. If I have a larger lambda value, then I will expect to wait shorter for an event to occur, and so on if I keep increasing the lambda value. So you can see that event here corresponds to these speciation events or extinction events that may occur in your tree. So what we want to do here is basically, we want to conceptualize age-dependent factor features into your rates, into your speciation rates. So I'll start by having these illustrations. So if you just consider you have a species, say species 1, and then species 1, basically you can consider that as a collections of populations. So you have like a population 1 of species 1, population 2 of the same species, all the way to say you have a finitely many population of the same species. Now these species are assumed to be like independent of one another, meaning that they don't interact with between each of these population groups. And what we want to find here is basically the first time that any of these populations diversify. So before I go further into this, let's talk about these distributions. So before you have the exponential distributions, this is described by this parameter lambda. Now you have these Weibull distributions. I'll tell why we want to consider these Weibull distributions. So Weibull distributions, in addition to having this parameter lambda, it also has this parameter k. So k is your shape parameter. So this shape parameter controls how your rates behave. So if you have like a k less than 1, it basically describes the rates that decreases over time. If you have a k equals to 1, it goes back to the exponential distribution. So exponential distribution is basically like a special case of a Weibull distribution where you have a constant rate over time. If you have a k larger than 1, then you may have rate that increases over time. Now, Weibull distribution is often used for modeling time to a failure of a machine, of a machinery. Now you can see that depending on different range that I put in the values for this shape parameter, I can basically control the rates as the time progresses. So this is important if you want to use these Weibull distributions to model the times to speciation events in your evolutionary model. So let's go back to this example that I have. So you have these n population groups of the same species. Now each of them have their own process, and then they are the same, but they are not interacting with one another. Now if I take the minimum time, so this T1, T2, and Tn is basically just like generic time where any of these species can diversify. So the minimum of all these T1, T2, all to Tn so they will be described. They can be described as a viable distribution. So you can see there is a relationship by having these kind of illustrations with and connecting it to the viable distributions. So now we have an age-dependent model that uses viable distributions. Now, can we have a more general age-dependent model? So why we want to do that? Because recall we want to basically improve our rate estimate. So we try to have a better model that can fit better to your data. So basically this more general age-dependent model, it still follows the age-dependent structure. So you can account for the age-dependent feature from your species in your into the rates, the speciation rates and extinction rates, but it still takes into account some hidden structures. So recall from before, in the earlier I said, no evolutionary models can capture the true underlying process that shape the empirical data, but we don't know what these unknown parameters are, but that doesn't mean you don't want to acknowledge having these unknown parameters in the model, basically. So we still want to acknowledge for that in the model. So what we do is that we consider a more general class of distributions. So this is called a phase-type distributions. I'll try to explain these phase-type distribution as clear as possible by using illustrations. So basically, it still models our waiting time, but it models the waiting times until a process reaching an immovable state. So what I mean by immovable state is that if you look at this diagram over here, this diagram consists of 3 compartments. You have the compartments that indicates the time where the process starts. You can move to a phase 1, to phase 1, but then from phase 1, it has some rates of moving from phase 1 to this immovable state. So what it means by immovable state is that once you are in the immovable, once the process is in the immovable state, it cannot escape from that state. So as an illustration, consider like this life and death cycle of process of a fish. So say fish start as a small fry, then it grows mature as it ages, all the way up to like it dies. So the death state of the fish corresponds to this immovable state of the process. So once you reach the state, you cannot escape from the process. So next, I'll describe how you can use these phase-type distributions to account for species age in your lineage diversification model. Now, there's also some interesting fact using this phase-type distribution. This is a theoretical result from 1996 paper that basically says any positive value distribution can be approximated using phase-type distributions. So this is good because times are positive. So times are always modeled by some positive values distributions. So this is the example of a diagram for phase-type distribution being used for lineage diversification. So let's say you have a new species that arose from a speciation event. Now, this age class, it definitely has a probability 1 of moving from this to the first age class 1. And then it can go to another age class, so it grows older and older, all the way to say age class n. And then from any of these age class, the species can undergo like a speciation event or an extinction event. So these immovable states. So these immovable state corresponds to a speciation event or an extinction event in your lineage diversifications. Now, what is good is that you can, if you see here from any of these age class, the process can escape to this immovable state. Now, if you can basically adjust these rates, these Q1, Q2, all the way to Qn to accommodate for different age-dependent models. So by having this kind of relationship, you can have a model with decreasing speciation rate as species ages. Or if you swap the order of the relationship between rates, you can have a model, say, with increasing speciation rate as species ages. And so you have a greater flexibility for fitting model in empirical data. And I'll show you that in the next slides that statement holds. So this is the paper that talks about this using phase-type distribution for age-dependent model. Now these are some results from it. So this goes back to this different way of basically measuring your tree shape. So your tree topologies and also the distribution of the nodes across the phylogeny that I mentioned earlier. So some facts from this model is that if you incorporate age-dependent feature for modeling your speciation process, it affects the topologies of your tree, of your generated tree. But if you assign the edge-dependent features into your extinction process, it affects the distribution of the nodes, internal nodes across your generated phylogen. Now, this is some model fitting to empirical data. So you can see that the first 5 model here, first 4 model here is basically, it has a lower data AIC. So it is actually a better model than already existing model that I mentioned earlier using the variable distributions. And you can also see these sub-model of this phase type distribution. This is the model where you have an age-dependent, an increasing rate of speciation as species ages. You can see it has a high AIC value, meaning this is a purely fit model to the data. So basically what the model says about the data is that you have a speciation rate that decreases as species ages, but these models that incorporate phase-type distributions still fits better than models that use a constant rate using exponential distribution or variable distribution that I mentioned earlier. Yeah. And you can also talk about these hazard. You can do the hazard plot. So what hazard plot does is basically it plots the speciation rates as a function of a species age on the x-axis. So you can see for that data, you have basically a decreasing trend for the speciation rate. So just as a reference figure, if you have a constant speciation rate, then you will expect something like this for your hazard plot. You basically have a flat hazard plot because you have a rate that don't depend on the species age on your x-axis. So just as a summary, so incorporating species age into the classification model can improve the rate estimates, speciation process controls tree topology under age-dependent model, and extinction process controls the distribution of nodes, and then it is a better model compared to other existing model for modeling waiting times to speciation under using the same empirical tree. Okay, so this is about age-dependent model. I'll go to another class of model that's slightly different, but still using the same concept. So this is the trait-dependent diversification model. So from before rates, we won't incorporate species age to play a role in your rates. Now we want to incorporate the inherited traits from your species to affect your speciation, extinction, and etc. So let's imagine here you have this hypothesis. Say, does the given trait play a role in a linear diversification for a given plate or empirical tree? So just an illustration, if I have like these 2 geckos, they have different colors. So you have like a yellow colored gecko. Maybe this yellow color corresponds to having higher speciation rate, but lower extinction rate. Or maybe the blue color gecko corresponds to having lower speciation rate and higher extinction rates. So basically we need a model that still accounts for how linear is diversifying over time. So having the speciation and extinction rates events, but still accommodates for how traits evolve along across your phylogeny. So how species transition from one state to another state. For example, this skin color. So these are some empirical studies that basically shows why it is important to incorporate traits, species trait into your diversification model. So this one is from 2020 that basically says the morphological transitions are the primary evolutionary driver for a class of fungi. And then you have another empirical studies that relates the pollination syndromes into and correlated with the elevated speciation rates in some phylogeny. So these are empirical studies. They use empirical trees. And what we want to do here, basically, we want to come up with a model. So this is the first model that incorporates the trait-dependent features into your events, your evolutionary events. So this is model from 2007. This is the binary state speciation and extinction model. So I'll explain briefly about what this model does in the next slide. So basically, you consider you have a species with discrete characters. So this character could be like I said, like maybe habitat types or like a skin color, like the colors that these geckos earlier have, or maybe it's like a food preference, etc. Now, for each of these characters, you can only have 2 states. So maybe moving maybe this blue colored gecko or yellow colored gecko, for example. So I'm using beetles in this case. Now, it assumes you have a constant trait-dependent diversification and state transition rates. What it means is that for each of these states, it has its own rates for diversifying, for speciating, and its own rate for becoming extinct. So you have this lambda1 and mu1 assigned to this color, and then you have lambda2 and mu2 assigned to the other color. And then you have these rates of transitioning between states. So you have this Q1, 2 going from color red to color yellow, or the other one, or the other way around. And then some assumption from this model is that events are independent because they assume so what it means is that if you look at this, so this is type of diversification that is not allowed. So you have basically a speciation events occurring at the same time with the state transition. So you have the transition from this black color to red color line. So you have a state transition occurring at the same time with the speciation event. So you cannot have two events happens at the same time. But you can have something that looks like this, where you just have like a speciation events without any state transitions, or you maybe have like a state transitions first and then followed by a speciation events. And then you have the identical processes across branches. What it means is that if you assign this distribution on your left-hand side, on your left daughter species, you also assign the same process for your right-hand species. So they evolve according to the same process, but independent of one another. So they are independent. These are some other trait-dependent models that people have considered. All right, so what's next? So as usual from before, when I introduced the age-dependent model, you start with a simple model, and then you try to come up with a more realistic model that hopefully fits better to empirical data. So this is exactly what it's trying to do here, is that we try to provide like an alternative and more general modeling approach to this earlier model that people have from 2007, so like these BC type models. And then along the way, we'll derive important metrics that are useful for studying linear diversifications. All right, so just start with the conceptualization. So this is what we call as a Markovian binary tree. So just as a diagram, what it does is basically it models the lifetime of a species until it dies. Now, during the lifetime of the species, what you do is that you have these events where the species can transition between different character states, like this red and yellow color. But you also, at the same time, count the number of daughter species arose from that ancestral species during its lifetime. And then this is the different way. This is the difference between the other model is that the expected waiting times is not exponential distributed; it's a phase type distributed. So like before, we have shown that the phase type distributions is better than just considering like a constant rates, like an exponential or variable distribution. So this one incorporates phase type distributions. So just some differences between the earlier model is that you have a more event types. So now from before with the BC, you cannot have a state transitions. You cannot have like this state transition happens at the splitting events, but here you can do it with the MBT. But it also accommodates like these different speciation modes that I mentioned earlier, where one lineage is the continuation of the parent's lineage, the other one is a completely new species where you basically just restart the process on that lineage. So just a bit of theory. So MBT is basically this Markovian binary tree. It falls under this theory that we call the matrix analytic methods. So as the name suggests, what it means is that all the model parameters are being captured in a matrix form. So all the computations are used in the basically matrix computations. So it has some great upside that I'll mention on the next slides. This is a more technical slide, but this is supple to mention. So we have like a phylogenetic trees, but we cannot see the extinct branches. What we see, we can only see the lineage that survive until the present time. So the present day species that we observe. So what we want to do is that we want to provide like a more careful treatment on how you actually view this extinction event, these extinct branches. So say if I have these two different phylogenies, this is the same phylogeny. The only difference is that this red dash lineage, it basically represents the extinct branch, the branch that leads to the extinct species. So the only difference is that on the left-hand side, the species that goes extinct happens after the ancestral species, this parent's lineage undergoes a speciation event. So just right after it undergoes speciation events, but on the right-hand side, it may occur, this extinction may occur before the speciation events of these parents of the parent species. So in that earlier model, these events are indistinguishable from one another. And then why it is important to account for this possibility? Well, say you want to know when the species actually went extinct if you have some sort of like an environmental shifts. Like, does it occur according to this scenario on the left-hand side or according to the this scenario on the right-hand side? And how does it affect our rate estimate, our extinction rate estimates? If you account for these possibilities of having lineage that goes extinct after speciation events and before speciation events of the main branch. Another way that MBT, this model can do is that it can compute tree likelihood. So this probability of observing the reconstructed tree given the model parameters without actually using the trait information at the presence. But it still assumes these trait dependent features, but it doesn't necessarily require to know this trait information. So this could be useful if you, for example, these trait data at the present are something that are difficult. Maybe it's not like a phenotypic traits that you can observe, maybe like a genotypic traits, for example. So these are some features that we also found along the way. So recall back, it relates back to these different statistics that we use to quantify topologies and then also distribution of nodes across trees. So rewind back to this tree balance. So yeah, from before you have this beta statistic. So the way they measure this beta statistic from your given tree topology is basically they use a statistical inference. So you have this, you compute this probability at each speciation time, you multiply them across these trees, and then you use like a maximum likelihood to find the best estimate for this beta parameter. So this beta parameter is used to basically measure the balance of a given phylogeny. So this is a statistical method, so it means that it's prone to statistical errors. So what we found with this model is basically you can actually compute tree balance directly from model parameters. So if you know the values of your model parameters from your model, you can actually compute the tree balance directly from it without having to use like any statistical inference to measure the topology. And this is just some results. So you can see you have this blue line and the red line, and they perfectly fit to one another. So red line is the one that based on the statistical approach, like using this maximum likelihood. The blue line basically based on the new approach that we have that computes rebalance directly from the model parameters. Yeah. And then this is also a technical slide. So we go back to these likelihood functions that basically defines the probability of observing a tree given your model parameter. Now this is just a snippet of the likelihood function under this model. So what this equation describes is basically these two possible events. So you say if you have a speciation event, you need to account for the way of placing the daughter species. Maybe you have the species one on the right-hand side following a speciation event, or maybe it's on the left-hand side. So this equation describes a speciation events and all the possible placing for these daughter lineages. Now, these more interpretable, you can interpret this, you can relate these equations with this actual representation of the tree above, but it has some drawback in terms of the computation. So if you do, it's very heavy in terms of computer memory. So if you look at this less familiar mathematical operator here, this is what we call as a Kronecker product. So remember that with the MBT, everything is described using the matrix computation. So you describe everything in terms of matrix and you do matrix computations. So with the Kronecker product, is basically you double the size, the dimension of your matrix each time you do this computation. So say if I have a 2x2 matrix that basically each row corresponds to each state of the traits that the species can have. And then if I do like a Kronecker product with another 2x2 matrix that comes from this species one over here, what you get as a result is basically 4x4 matrix. So you can see the size of your matrix for each of these computations, it gets bigger quadratically. So it's not very efficient in terms of memory, but we have a way around for that. So this equation at the bottom, this is basically the same. This is the equivalent representation of this equation on the right, but this is less interpretable in terms of how you relate this equation with the actual representation of the events above. But what is good with that is that now you have another less familiar mathematical notation here. This is what we operator. This is what we call as a Hadamard product. This is another matrix computations. But now with this Hadamard product, you basically preserve the size of your matrix. So if you have a 2x2 matrix and then have a double product with another 2x2 matrix, you still get a 2x2 matrix. So you don't get like a 4x4 matrix as you do with the canonical product. Yeah. So just a summary. So species trait could explain some behavior of your speciation and extinction rates as seen from your empirical data. Now, this Markovian invariant tree is an alternative approach to the already existing trait-dependent model and possibly more general model. And then you can actually do another stuff with that. You can basically compute them. You have another alternative way of actually measuring the balance of your tree directly from your rate parameters, and also an efficient algorithm for computing that likelihood function that I explained just recently. All right. Sorry for the long talk. This is the last part of the talk. So we have a model that incorporates species age, and then you have a model that incorporates traits. So you have the species characters. Now you want to have a model, the linear diversification model that accounts for species range evolution. So how the range of species evolve over time. So I have this figure here. So for example, why do species live where they live? So I have a picture of a Komodo dragon here. It only exists in a few islands in East Tenggara province in Indonesia. So why Komodo Island only exist in these few small islands in this particular part of the world? So how do these sequences of speciation, extinction, and dispersal events relate to these present-day biogeographical patterns that we observe? So this is just an illustration of a phylogeny under like this diversification model for species range evolution. So this is a model from 2011. This is the GSC model. So what you observe at the present is basically the species ranges. So say if you only consider two regions, say two discrete regions A and B. Now maybe this species only occupies region A, so this is an endemic species, or maybe it occupies all the regions that you consider in your model. Like this species over here, now this is a widespread species. So what you observe at the present is basically all the different ranges of your species that you consider in your study. What you do is that you start assigning rates to even patterns. So I have an illustration here. So let's consider like a simple model with two discrete regions. So you have a red color region and a blue color region. Say you have this blue angry bird species here in region color A in red regions. Now it may disperse to another regions. So it expands its population. So now it also occupies in this region over here, this blue region. Or maybe it has like a within region species speciation. You have this blue bird in the red region. You have this different species in another regions. Now you have a within region speciations in these particular regions. Now you have two different species that occupies the same region. So you assign this rate WI for that regions. Or you maybe also have an extinction event. So for example, at some point in the future, this red species went extinct. So it disappears completely from this region. Now, because this is an endemic species, it only occupies one region. The branch for that species, the lineage for that species basically becomes extinct. Or you have another type of speciation, what we call as a between region speciation. So maybe you have like a widespread species, this red bird that occupies both regions, but there is no barriers that separates these two regions. So you still have like a gene flow moving between these regions. But maybe at some point in the future, you have like some sort of like a physical barriers that separate these two regions. So there's no interaction between these. And so you now have two completely new species, each occupies their own regions. And so what we want to try to find with this research is basically so what we can see is basically how the species ranges at the present time, right? So we can calculate the frequency of species that occupies only, say, region A, species that only occupies region B. We can categorize them based on their, say, maybe widespread species. So these things that you can see in the present. So what we want to find is basically we can try to link that observation at the present with different scenarios that might correspond to the patterns that we see at the present. So maybe you have this scenario one where you have a larger region A and smaller region B, and then they are further apart, so you have a lower dispersal rate. Now, if you have a larger region A, maybe it corresponds to having higher speciation and lower extinction rates, and opposite can be said for the region B, or maybe this observation can be explained by scenario 2, where you have a smaller region A but larger region B, and then you have a higher dispersal rate between the two regions. And now this has a different interpretation in terms of its speciation and extinction rate. But maybe both of these scenarios could explain this observation, this present day observation that you see in at the present. So, so what we do is basically borrow the theory from population genetics in the macroevolutionary context. So diffusion approximation is commonly used in population genetics to model dynamics of how allele changes, allele frequencies changes in a random population. So say if I have like a right Fisher model, so this model from population genetics without mutations. So you have like these two different allele types, and you consider like these discrete non-overlapping generations, and you basically want to find, say, expected waiting time for one of these allele types to become fixed in your random populations. So so what we do is that, so this paper here, this is the paper that links this theory from the population genetics to study the macroevolutionary dynamics in that state-dependent model that I covered earlier. So what we actually do is that, so you have this forward in time process. So your y-axis is basically, it counts the frequencies for one of the range states that you consider, and then time moving forward from the left to the right. And then you basically want to simulate this dynamic of how the frequencies change over time. So it's basically forward in time simulations. It's defined by this equation here. So this equation has this stochastic component described by this is a sigma notations, and then it has this deterministic components. And what you do is that you basically want to compute the future value. So this pi I at t plus delta t using the current value. So this pi I of t. So you basically want to find the expression that describes this deterministic component and the stochastic component, and use the equations to basically simulate the trajectory over time. So in general, what we want is that we have this observed state frequency. We use this mathematical tool and then to link between different biogeographical scenarios with the observed state frequency that we see at the present. And then this is a sum result. So there are actually two lines here. So you have these red lines that comes from this diffusion base, and then the other one is the frequency that you count by simulating trees. So you simulate your trees under some parameter values, and then you basically count the frequency for each of these range states. And these blurry lines here is basically just it's a different replicates of the simulation based on this diffusion process. So you can see they perfectly fit within one another. So you can use this approximating model via this diffusion process to basically predict how the range state evolve over time. All right, so another result that we have is basically the existence of multiple rate scenarios that leads to the same stationary frequency. So recall earlier from illustrations, you have an observed state frequency, you have a different scenarios. Maybe both scenarios could explain what you see at the presence. So this is what exactly we have. So we have these two different figures, two different dynamics, but they represent the same stationary dynamics. You can see these red line, they converge to the same value as the one on the right-hand side, same with the blue one, and then the purple ones. But they all are evolved according to a different rates. So the one on the left, you have a lower within region speciation rates, and lower dispersal from region B to region A, and lower extinction in region A. And the other one, you have a higher within region speciation, higher dispersal from region B to A, and then higher extinction rate, but they all converge, they all go to the same stationary frequency, the same observed state patterns, the range state patterns that you see at the presence. So just as a summary, so you have this stationary frequency, you start with the stationary frequencies, things that you observe at the presence, you assume to have in the presence, and you have some models, you have the diffusion models, you find from this model, you get the rules that basically describe the relationship between rates in your model. If you have these rules, you can basically perform random draw from these rules to get the rate values from that model. So you start from the stationary frequency, you can get the rates that correspond to the stationary frequency, but you can also go backwards. So start from the rates values, you can get a theoretical, like a stationary frequency out of these rates. So this is like a projections mapping from stationary frequencies to rates relationship that you hope to see. So this is some further reading. This is a preprint that we just have. Further, and just a summary, you have this general framework that studies how state dynamics evolve over time in the model that accounts for species range evolutions, along with the linear classifications. And then we saw from simulation that the approximating model works as good as the true model, the true generating process, for example, this GC model for simulating state dynamics. And we have this one-on-one mapping between rate patterns, actually many-to-one mapping between rate patterns and the observed state frequencies at the tips. So just moving ahead from this work, what we can do is basically, we can do some like model adequacy tests. So like this paper here is basically, they perform adequacy tests in the state-dependent model by using like a simulation-based approach. So what we can do is that because we have this theoretical link, this theoretical result that link between stationary frequencies and the rates relationship, we can actually validate that what we see from the empirical data match with what we hope to see from the model. Yeah. And another consideration is that, so these rates that I described earlier, they are constant over time, so they are not changing over time. So all you can do is that we can introduce this notion of time heterogeneity. So maybe you have this kind of different scenarios. So maybe at the first half of the period, you have this kind of scenario over here that corresponds to this specific behavior. Maybe at some point in time, you now have like a region shift or region changes, and then now you have this kind of scenario that also leads to the different dynamics of the state frequency. So the question that you want to ask is that how does the evolutionary systems re-equilibrate in the presence of these rate shifts in time? Okay, so that's all. If there's any questions, feel free to ask. Thank you.
So much. I can take any questions.
So for these models, for example, the age-dependent model, do you need to like take a lot of other people's data to train the model to get all the factors to work? I'm not.
So the way it works is that basically if you have a model, you can have this likelihood function, right? So you have these likelihood functions that basically calculates the probability of actually seeing these empirical trees that people already generate. For example, people already have this empirical tree. So you already have this tree. Now you have the model. You have these likelihood functions, this probability function that you derive from your model, and then you perform like a statistical method to actually infer rates from your data. So basically, what you need is basically just these branch lengths, this length of these branches that you have in your phylogenetic tree, in the empirical trees that you have to actually like infer rates from the data. Yeah. The rates, yeah. So like the speciation rates or like the extinction rates, for example. So this model assumes you have a phylogeny because like you actually have your empirical phylogenies from maybe like a genomic sequencing, for example, and then from the model you can get the rate estimate out of it, like what the speciation rates, whether the speciation rates from your tree maybe tend to decrease or maybe increase over time, for example, or maybe not in the overall tree. Maybe just consider like a specific clades in your tree, and you want to examine these specific clades on your tree. Yeah. That's not sure, because like all these models basically like you assumes that you already have this phylogeny with all the so with the trait dependent model you have these traits traits data that basically assign at the tips of your phylogeny. So you observe these trait data at the tips, and then use. The idea is basically you have these functions, these likelihood functions that you get from your phylogeny, that you get from your model, and then you already have your data with all the tip state informations at the present that maybe other people already derive from their empirical studies, right? And then you get this rate estimate, these speciations, extinction, and also these rate transitions, these transition rates. Yeah, but actually, like this MBT model, like the one that I just presented, you don't actually necessarily require this rate information at the present to actually start this likelihood computation. So basically, what you do is basically, if you have a phylogeny, you start from the tips of from the bottom of your tree, so the present time, and then you just sort of like a transverse towards the pass to the top, and then you have the product of all these different probabilities that are assigned to like branches that I described earlier, and then use like a statistical inferences, maybe like a maximum likelihood, to find like the best fitting parameter values to the given your data and then your model parameters. Yeah, hopefully that helps. Yeah.
Thank you. Really cool talk. I appreciate you sharing that. I'm curious with the age-dependent diversification model, that's taking into account like absolute time, not generation time.
Yeah, it's absolute time. Yeah.
Absolute time. And so model that you like changes in generation time to sister lineages. It seems like if on one branch you have say fluctuating generation times, right? They're experiencing like more time. Are those confounded or?
Right. So basically, all these models, they assume like an independence of branches. So branches don't interact with one another. So all the process, like say you have say a phylogeny with say just two species. So you have like three branches, right? So you have these ancestral branches and the two daughter branches. So the process that you assign, the age-dependent process that you assign on the ancestral branches, ancestral branch is the same age-dependent process that you assign on the left branch and also on the right branch. And depending on what sort of like a speciation scenario that you consider, like this symmetric speciation, basically what it does is that every time you have a speciation events, you reset the process. So you assign the new age for the descending species. But if you have if you consider a scenario with these asymmetric speciations, you keep the process for the left, say left lineage, but the other one, the daughter lineage that descend from that ancestral species, you just reset the process back to zero. But you keep assigning the same age-dependent process into the model. So these independent of branches, it is assumed because the way it works, like you have these different probabilities assigned on each branches, right? So if you have independence, you can basically just multiply them together. Now, if you can multiply them together from the roots, from the present of your phylogeny towards the roots, you come up with this likelihood function, this probability of observing the whole tree, the whole reconstructed tree.
Sure. Thank you.
Yeah, thank you very much.
Question: for the trait-based approach here, does that allow for any interspecies variation traits? So, or do you have this absolute monophily everywhere that the trait is just one by each species?
Yeah, so this is like a model with discrete traits. So you assume the traits to be discrete, like for example, like this say food preference, for example. But you can have multiple states for one character. So maybe you don't just have like two different food preference. You can have like three different food preferences, etc. And then you have these transition rates that basically species can move from one character state to another character state along its lineage. Yeah, all the way.
Okay, so but with that allow for like reversion rate, so like where they move from.
Yeah, so it can reverse. So yeah, so you have the rates of going from say character state 1 to character state 2. or you can move backward from character state 2 to character state 1. So actually like see if I can, hold on. Like these yeah, so like these this illustration here with the beetles, like the colors, like you have these, so each of these character states has its own rates for speciating and extinct, but it also has this transition rates for going from say yellow to red and then going backward again from say red to yellow.
Folks have more questions, we can chat at the reception that will take place just across the hall. And please join me in Albert.
Thank you very much.